Who would have ever thought we'd talk about lines of therapy and sequencing of therapy for patients with gastric cancer? But you know, it's interesting. I'll also ask you, now that we're looking at some new target agents, a lot of clinical trials are out there, this sort of gestalt that tumors change over time, do you all ever repeat a biopsy after the patient's gone through second line of therapy, for instance, before third line? David, do you?
I think in the context of a trial. I think outside of a study, I think there's limited value for doing this. I think this is why we incorporate biopsies into research trials, because we want to learn about are there acquisition of new mutations and abnormalities. We want to have a more up-to-date characterization of the tumor. We do know from our own database within Memorial, looking at trastuzumab refractory patients, when we rebiopsy them, they acquire all these new mutations and they lose HER2 expression. But I think, and now with the advent of potentially liquid biopsies, doing blood-based analyses needs to be explored to avoid the morbidity and invasiveness of repeat biopsy. But we're now in an era where there's a lot of active drug development, certainly trying to develop. I think we've kind of hit a plateau with checkpoint inhibitors, and I think we really need to look at novel combinations. How can we make a cold immune environment hot? Are there ways that we can augment checkpoint inhibitors? Are there novel immunosuppressive mechanisms that we haven't even developed yet? Can we combine some of our existing agents like PARP inhibitors that may augment immune microenvironment, even drugs like ramucirumab and VEGF targeted agents? I mean, Kohei, you can comment on your rather striking results with the regorafenib, a tyrosine kinase inhibitor targeting VEGF and checkpoint inhibitor therapy.
Yeah. So the trial is just a phase 1 trial. We reported the result in this ASCO, which includes 50 patients with 25 gastric and 25 colorectal cancers. Overall, we observed 40% response rate, 44% in gastric and 36% with colorectal. So from the safety point of view, initially this is a usual 3+3 design, but we decided to finally 80 milligram of regorafenib is the optimal dose because of safety. And from the preclinical point of view, there's very interesting data which support this lower dose of regorafenib. Our concept is to target immunosuppressive cells such as the Treg cell and the tumor-associated macrophage. If we use a higher dose of regorafenib, not only TAM but also M1 macrophage and the CD8 number reduced. So still, we reduce the dose based on safety profile, but still support, there's this data support a lower dose of regorafenib. And actually paired biopsy before and after treatment showed a clear reduction of Treg cell, which may be in line with our concept. And currently we are discussing about the future steps.
Well, we already have phase 2 data for regorafenib in refractory gastric cancer, that it may have some disease stabilizing effect, but no responses. So to actually see a response in a combination is a signal. And there is an ongoing randomized trial of regorafenib versus best supportive care in refractory gastric cancer. And another VEGF tyrosine kinase inhibitor, ipatinib, is actually showed superiority in China. It's actually approved in China as a late-line treatment option.
Yeah. I think it highlights what we're talking about, is that, you know, immunotherapy has modest activity in gastric and esophageal cancers, really 10 to 15%, even if you're PD-L1 positive. And we're doing a lot of work, and you know, Kohei's, your study is a fantastic example of that, to try to combine drugs to improve the efficacy of immunotherapy.
And so tell us also a little bit about, so we're doing new combinations of immunotherapy. You know, Kohei, you do a lot of phase 1 work. Outside of VEGF inhibitors, what are you seeing now? What agents are in play for combination immunotherapy?
Yes, so I do several trials, but first concept is to target immunosuppressive cell, like a previous regorafenib trial. Other trial I have conducted now is a heat shock protein 19 inhibitor, which also reduce the number of immune suppressive cells. We confirm this phenomenon in preclinical study. And I also do a CD4 depleting therapy, which is a, target all immunosuppressive cells. It shows some single agent activity. But these targeting immunosuppressive cells may not enough for all patients with gastric cancer, especially as mentioned before, cold type of tumor. These tumors usually have a low infiltration of CD8 cell as well as the immune suppressive cell. So maybe additional treatment to kick the immune system might be important. One of the candidates is oncolytic viral therapy. We previously reported preliminary data in esophageal and gastric cancer in previous AACR. It showed some activity in primary tumor as well as a distant metastasis. And the local tumor showed the infiltration of T-beta high activated CD8 cell. This is one candidate. And the other is we will start in the near future other local therapy. I cannot disclose today, but the other local therapy in combination with immunotherapy, this trial will be started soon. And additional cell therapy or CAR therapy may become additional breakthrough for patients with cold type gastric cancer in the future.
Yeah, it's super exciting what's happening with immunotherapies, the bispecifics, the BiTEs, the CAR-Ts, the combinations. You know, it's interesting, people then go back to think about gastric cancer, and everybody always loves that age-old combination of PD-1 inhibitor plus CTLA-4 inhibitor, David. Nivo-Ipi, gastric cancer.
Yeah, I think the original sort of comparative phase 2, comes from one of my colleagues at Memorial, Yelena Janjigian, and combination Ipi-Nivo was chosen as one of the randomized arms in upfront therapy because of a response rate potential advantage. But even from the outset, the signal wasn't that strong. There really wasn't a clear difference in progression-free or overall survival for nivolumab alone versus ipilimumab plus nivolumab, and it's a toxic combination. I know our melanoma doctors use it all the time, but that arm was actually dropped from the first-line randomized trial because of adverse events. So I think at the current time, the combination of CTLA-4 and PD-1 is kind of on the back burner. I mean, there are some alternatives. Tremelimumab as a CTLA-4 drug is still being looked at in phase 1 and 2 trials. But they're likely going to be better partners because it's going to be hard to imagine adding something else to ipilimumab and nivolumab. And without a clear signal with adverse event rates, it's probably not going to be something that's going to move forward.